Okay, this is the third recording for Chapter 7. Hopefully we'll end, this will be the last one. But transport across the plasma membrane can be hard to understand. But I would say before we review passive and active, let's discuss the substances that move across the plasma membrane directly through the phospholipid bilayer. So versus the ones that need a membrane protein. All right, so the substances that can move through the phospholipid bilayer by simple diffusion, just diffusion, simple diffusion or diffusion is fine. Those are going to be nonpolar substances, and a lot of times they're also small, but it can be any kind of lipid or fatty acid, steroid hormones because they're lipids, and it can also be the small gases that are insoluble in water, carbon dioxide and oxygen. Okay, so now we talk about substances that move by passive transport, which requires no energy, versus active transport, which requires ATP. So passive transport is just when molecules move from their high to their low concentration, down or with their concentration gradient. You can see all of these molecules are moving toward their low concentration. Okay, so they're going to move wherever their concentration is low, that's where they're going to go. So the orange triangles are moving through a channel protein, and then the blue rectangles are moving, being transported across through a carrier protein. Either way, it's called facilitated diffusion. With active transport, you also have a carrier protein involved, but it's going to be two differences between active transport and facilitated diffusion. One has to do with the direction. Molecules are going to move toward their high concentration against their concentration gradient. They're going to move wherever their concentration is already high. And the second difference is ATP is required. Okay? So that's passive and active transport. There's a section here on the membrane potential or the voltage across the membrane that I'm going to skip. But I will tell you that sometimes a substance uses cotransport to move across a membrane, and I can explain it better with a picture. All right, so active transport was used here in what's called the proton pump. Remember, hydrogen ions are protons. So this proton pump is pumping hydrogen ions or protons toward its high concentration. The purpose of the proton pump is to build up a lot of hydrogen ions outside the cell. In other words, to increase the hydrogen ion concentration outside the cell. Okay, so it's creating a really big, a really large concentration gradient where there's a lot of hydrogen ion outside and not very much hydrogen ion inside. Now, since it is a pump, and it's using active transport, and it's transporting toward the high concentration against the concentration gradient, then it's active transport and it requires ATP. So that would be an example of active transport. But then when the hydrogen encounters a transport protein, a carrier protein, that will allow it to diffuse or to move by passive transport, it's going to naturally diffuse from where its concentration is high to where it's low. So when that hydrogen moves back into the cell by passive transport through this cotransporter protein, sucrose is also allowed to move into the cell using the concentration gradient of the hydrogen, using the movement of the hydrogen. So that's cotransport. You see that sometimes. This is a good example. It's talking about diarrhea, but it could be diarrhea, it could be vomiting, anything that causes you to lose a lot of body fluids. It's going to also cause you to lose electrolytes. So when a person has diarrhea or does a lot of vomiting and loses a lot of body fluids, their sodium levels can drop so fast that it's life-threatening. There's a disease called cholera, which is basically diarrhea. You just have so much diarrhea that you die. And it's not just people. Little puppies and little kittens can get, what's that called that they can get? Parvo. They can get parvovirus, which causes severe diarrhea. And unless you're able to just basically hook an IV to them and replenish the fluids that they're losing, or get them to drink like I know some who have had their lives saved just by feeding them like Gatorade, or something like that, or Pedialyte, something that has electrolytes in it. But anyway, diarrhea can kill a person, or can kill an animal. So if you drink a concentrated salt and glucose solution, like, your sports drinks, Gatorade or whatever, Powerade, then the glucose can be taken up through a sodium glucose cotransporter, and that could save a person's life when they're having diarrhea. Okay, the next thing is we're going to go over bulk transport, and that's endocytosis and exocytosis, and you have an animation on slide 90. But bulk transport means that the amount of substance that's coming into the cell or leaving the cell is so large that it cannot diffuse, it cannot move through the phospholipids or through a channel protein. So it's just a large amount of material. And if the material is being transported out of the cell, then we call it exocytosis. A transport vesicle inside the cell basically contains a substance to be released outside the cell. So it moves to the plasma membrane and fuses with it, and then opens up and releases its contents outside the cell. So exocytosis uses transport vesicles to move substances, a large amount of substance, outside the cell. And a lot of times, cells that secrete something, like the cells of the pancreas secreting insulin, they do this by exocytosis, okay? So endocytosis, and well, this is a good picture to look at. So your insulin molecules, let's say, are these little blue molecules. The Golgi packages up the insulin into a transport vesicle. The transport vesicle, look at it, moving along a microtubule or a track. That shows you a good example of using the microtubules as a track to guide vesicles. So anyway, that vesicle moves to the plasma membrane, fuses with it, because the vesicle membrane and the plasma membrane are both phospholipid bilayers. And then once it fuses with the membrane, it opens up and the substance, like the insulin, moves outside the cell, enters your blood and travels through your blood to your liver cells. Now, endocytosis is a little more complex because there's 3 types of endocytosis. But basically, whatever is coming into the cell, the plasma membrane forms a vesicle, a pocket around it, and then it encloses and forms a vesicle around it. And then the vesicle is brought into the cell. So there's 3 types of endocytosis. There's phagocytosis, and that is cellular eating. That is when the cell engulfs a solid substance. It could be a white blood cell phagocytizing a bacterial cell or a germ. It could be an amoeba that we saw earlier, an amoeba in chapter 6, I think. An amoeba is a protozoan that forms pseudopods and wraps around the food that it's going to eat, and it engulfs its food by phagocytosis. So phagocytosis is cellular eating. It's bringing into the cell through a vesicle some solid substance. Then penocytosis is cell drinking. It may not just be water, but it can be a solution, but it's something liquid. That is a large amount that's being brought into the cell through a vesicle. And then we have a specific type of endocytosis called receptor-mediated endocytosis. And that is when the substance that comes in has to bind with receptors on the plasma membrane surface. So we're looking here at possibly an amoeba with pseudopods, forming pseudopods and wrapping them around the food particle that it's engulfing. This is phagocytosis. So the food particle, basically the plasma membrane forms a pocket around the food particle, and then it eventually encloses the food particle and comes into the cell. This is a food vacuole. In penocytosis, same exact thing, but the plasma membrane encloses around a solution and then pinches off and brings the solution into the cell through a coated vesicle. Then we have receptor-mediated endocytosis, and that's when the plasma membrane has these receptors for specific substances. LDL, which is low-density lipoprotein, we also know it as the bad cholesterol. There's good cholesterol and bad cholesterol. But the bad cholesterol comes into our cells this way. It comes in through these receptors. And so that's receptor-mediated endocytosis for something specific. All right, phagocytosis, penocytosis, and then receptor-mediated endocytosis. I was looking to see if, they do. They talk about the LDL receptors. And so cholesterol accumulates in the blood. And so if a person doesn't have these receptors, like if they have a genetic disease, familial hypercholesterolemia, which is listed right here, I just saw it. That's just genetic high cholesterol. So in that case, it doesn't even matter what their diet is. It's hard to even control this condition with diet. They have to take cholesterol drugs. Typically, they're drugs called statins. But anyway, they don't have LDL receptors, or if they do, they're defective. So they're not able to remove that cholesterol from the blood and bring it into the cells and store it. All right, there is another bioflix on membrane transport, and that is slide 103. All right, and I think the rest of these are just extra slides, so I'm going to stop our recording right here with chapter 7.